Hi, everybody. I am super excited to have you all join us this summer in Textile 2023. Thank you so much to everyone who is able to make it to our live panel. To those of you unable to make it, I will be uploading it within the next week. Today is going to be our first module on an introduction to data science. I'm going to do a brief intro to data science in the Nance lab with some interactive activities, followed by an introduction to a data scientist role, and finally a small intro to Python and working with Google Colab, which is where we will be doing a lot of our coding this summer. In a lot of our presentations, you'll see this slide as our beginning format. So typically we have some title, who created the slide and the departments they belong to, the name of our lab, which we either go by the Disease-Directed Engineering Lab or the Nance Lab, University of Washington, and then just some details about the program. The way that I structure my slides, which may be different than how Nels and Foon structure their slides later this summer, is that I often have a title at the beginning. I include an explore more box off to this side of the screen here that'll often have a meme or a picture, as well as a QR code to learn more if that's a topic that interests you. And then at the bottom here, that will go away when I stop using my mouse. Here, let me use a highlighter. At the bottom here is our toolbar, so you can kind of see where I'm at in this presentation. This presentation split into 3 chunks, so just keep that in mind. Since this is an asynchronous module, please feel free to pause this whenever you want to, and you can take some time away and you can come back to it. There's also a Google form you can save as you go. The title tells you what it's all about. The side plays for memes, comics. This is so you can refresh. And this toolbar is just to keep track of where we're at in the module. At the University of Washington, we like to give land acknowledgements. So the University of Washington acknowledges the Coast Salish peoples of the land on which we teach, learn, and study, the land which touches the shared waters of all tribes and bands within the Duwamish, Tulalip, Suquamish, Tulalip, and Muckleshoot nations. Since this is our first time doing a land acknowledgement, I just want to provide a little bit more information about them. So why do we do a land acknowledgement? An acknowledgement is a simple, powerful way of showing respect and a step towards correcting the stories and practices that erase Indigenous people's history and culture, and toward inviting and honoring the truth. This is a statement from the U.S. Department of Arts and Culture. There are QR codes on the right here. My highlighter has disappeared. QR codes on the right here. Wow. I didn't know that. I meant to pick a laser pointer. Thank you, everybody, for bearing with me. QR codes on the right here if you want to learn more about Native land from the Native Governance Center or UW-specific resources on land acknowledgements. From this guide in the upper right corner, you can get great things like a self-reflection on why do you include a land acknowledgement, some places to do your homework about the Indigenous peoples to who the land belongs, history of land and related treaties, and how to use appropriate language, such as how to use past, present, and future appropriately. If you'd like to see the native lands that you live, learn, and work on, there's this fantastic website that's interactive, and I included the resource here. So, this summer, you're going to be taking part in TEXTILE. TEXTILE stands for Tutorials and Experimentalist Interactive Learning. In a way, I kind of picked that because I wanted this acronym to be related to my other acronyms, but it's just a scientific training program for data science-based research from real biological data that we include in the lab. So how this works to me is that we have some lessons that are research lab specific. So this is what we're going through right now, which is data science in the Nance lab. And then we have some data science specific lessons where we're going to go straight into a data scientist role and kind of skip this brief history of coding due to some time constraints and things that we chose to add on at the end. And then we'll also get into image processing specific work. I'll introduce some of the scientific applications of that today, and each of these are an individual lesson that will build off of the data science, build off of the wet lab. This is from when I published this work as a paper when we first taught it. We have added on wet lab portions and machine learning portions that Foon and Nels will teach respectively. So I will teach all of my work first, and then I'm moving to Baltimore. And so in that process, Foon is going to come in and actually teach you about the wet lab work. And so you'll get to understand how we generated the data you're working with in my section. And then Nels will come in and teach you some machine learning. And I will finish out your final week by kind of wrapping that all up together into a cool project and doing some reflection. Here's a paper here about training the next generation of physical data scientists, which I think is pretty cool. So this is a common misconception that we get from the textile program. Textile is about coding. Why aren't we doing more coding? And so the way that we approach data science in this program is that data science is a combination of computer science, math and statistics, and domain expertise. So if all you want to do in your life is code, I highly recommend that you become a computer scientist and you study computers. But if you want to be a data scientist, then you need to learn all three of these aspects, which doesn't necessarily occur just by coding. For sure, we will have entire coding modules, but I just want to frame this context for you. If you're interested in learning more, we highly recommend Free Code Camp, which will really dive into the principles of Python that we're teaching you here. But you'll learn enough coding in this class to do what you need to do, so you don't have to worry about doing outside education unless you're interested. So how it typically works, this is how our modules are kind of broken down. We often have some pre-module activity. Today is a little bit different because it's your first intro. Then we have a main module, that's what you're going through here. And then I have a Google form which has a post-module reflection, and I will send out feedback forms as well. And then we have in that main module some type of presentation, an interactive session, a main presentation, an interactive session, a concluding presentation, and a reflection session. The way those interactive sessions work will change based on, is it a wet lab module, is it a coding heavy module, is it a data analysis heavy module, all of those types of things. If you want to understand why I chose modules or you're interested in educational research, there's a fantastic paper over here that talks about modular learning, a way to strategically plan education so that people retain more information and are more engaged. There's a funny lesson in this meme up here, which is talking about just getting students to engage. So the mother goes, okay, students, who knows the answer? The teacher, and then the mother goes, speak up, and the student doesn't want to, and interaction is important. The schedule, which I posted for you all, kind of looks like this, unless there is a date off, such as Monday. So often I'll post the paper of the week on Monday with some questions that you'll respond to in Slack. I'll respond to all of those over the weekend, so I prefer that you post them on Friday. I love responding to you all. I love engaging in conversation, so please participate in those. And then the poll of the week opens Tuesday, and this poll is designed so that you begin to think about things in the domain science or data science side that you may not have thought about before. So you'll probably have seen this week's poll, what's your favorite brain cell? I just want you all to think about different types of brain cells. I didn't know more than neurons existed really until the second year of my Ph.D. when I began to focus on microglia, which we'll talk about more. Mondays and Wednesdays, we upload our modules. Tuesdays and Thursdays, we have some type of panel or workshop. And starting tomorrow, we have asynchronous office hours. So tomorrow will be our first experience with that process. If you're interested in why I don't have a lot of grades, this entire program is participatory. Here's a fantastic education paper about teaching more and grading less. You're really going to get out of this program what you put in. So if you put in a lot of discussion and a lot of time, we will put time into you back. We will get to know you. We will help you on your career journeys. We will talk about research. And if you don't put in a lot of time, you're going to get less out. So this is just really about that. It's not meant to be a part of stress. It's not meant to be a part where you're going to fail or you're going to pass. It's meant to let you explore if research in a neuroscience related field or a cell bio field or data science is something you're interested in for a career. And I want that to be stress-free. So who is the Disease Directed Engineering Lab? This is a one of our lab meeting photos. There's quite a lot of us. I love this picture over here in the corner. I'm now a postdoc, so I'm back here, which is like showing the meme that occurred during COVID when the kids broke into this news person's room, but the professor has it all together. The PhD student is a slightly older kid, the undergrads are the babies of the lab, and the postdocs are trying to keep it all together. In our lab, we have our principal investigator, Dr. Elizabeth Nance. She's our professor. She's the big boss of the lab. And then we have graduate students. We have only PhD students right now. You'll see Nels, he transitioned as a master's student. And then we have research trainees, which are undergraduate, 2-year college, or high school students, which are typically mentored specifically by these different graduate students. There should be a third arrow here of not leaving off the short hair presenting graduate students from that group. Academic research is often highly independent, but it is more successful with a team. So you often work by yourself on a project, but in high collaboration with other people. So you have your project, you're really steering it, you take ownership of it, but you're going to talk to a lot of people for help. A lot of people are going to train you on equipment. A lot of people will have reagents that you need, or training that you need, or insight that you want. So what do we do? I will have you explore the Nance Lab website in just a few minutes, but this is how Elizabeth, Dr. Nance, really breaks down what we do. On one side, we quantify and predict how nanoparticles transport, compartmentalize, and where their fate ends up in the brain. Nels will be talking more about this with machine learning later. On another side, we engineer effective nanotherapeutics for neurological disease. Foon will be talking about this a lot with her extracellular vesicle work. And then we also probe developmental and disease processes to elucidate insights into structure and function of the brain, and develop robust, unbiased, quantitative fluorescent image analysis pipelines. All of our module applications are to probe developmental and disease processes, but I'll be teaching a lot on this developing robust side. So you'll really get a lot of different aspects to the Nance Lab through the summer program. So Nell's machine learning, Foon's wet lab modules, and my data science modules. And then I just want to give you an example acknowledgement slide from my thesis presentation to show you just how complex academic research really is, that even though it's independent, no man exists as an island. So this is the slide I included on my thesis. So you can see all the graduate students we have in the lab. Everyone bolded on an acknowledgement slide from our lab contributed to that presentation. That we have a lot of lab alumni who have been a big part, that as grad students we get thesis committees, we have a ton of amazing collaborators, we have a great set of undergraduates, and we're also really thankful for the animals that we use and the data that we're able to get. So this is just an example to give you an insight into research. So how do we fit into the greater UW community? So these buildings are a lot of engineering over here, with this being BioE, and Benson right here is chemical engineering, and we are on the second floor. If you want to learn more about UW Chemi research, not just us, there are other amazing bio labs, but also materials labs, polymers labs, computational groups, and like energy and synthetic bio. I highly recommend checking out that QR code. UW Chemi really fits into a bunch of areas, but Elizabeth and her research fits into so many. You probably think of us most as health and biotech, but we also do a lot of data science, and we had some students who did molecular simulation who since graduated, but Elizabeth could return to that. And we also do a lot of advanced materials development in our nanoparticle team, which will have the least amount of interaction with, but they will show up on some panels later. This is wrong. Please attend our Meet the Principal Investigator Q&A session next week from 4 to 5 p.m. to talk to Elizabeth Nance. Sorry about that slide. I'll fix that. So this is our first interactive session, number 1, explore the Nance Lab website. So this Google form here that I'm showing you is going to work for the entire presentation. Please pull that up. Pause this video, go to the QR code and the Explore More, and answer the questions that are in this first part. The purpose of this interactive session is I want to get you all used to exploring research lab websites. I want you to begin to see the components that exist of research labs so that when you leave from our program, you feel more equipped to apply to other research programs, or you understand the infrastructure of labs a little bit more so that you might have a leg up in your process. So please pause here until you complete that first section, and when you go on to the next session, return to the video. Great. I'm so glad that you did that and you got to know a little bit more about the NANCE lab. I hope you feel a little bit more confident in navigating lab websites. This will come up, especially in one of our panels later, and it will come up later in your life if you're interested in research. I've explored so many lab websites. So now I'm just going to give you a brief primer into the domain science side of what we'll be doing for the summer. So what we're going to look at this summer are immunofluorescent images of microglia. Don't worry if all of that is brand new to you. We will be introducing it piece by piece, but I just want you to see some things here. So immunofluorescent imaging is the way of attaching dyes to cells and imaging them with a high power microscope, and that makes them show up fluorescently, and we can take images of that. We specifically use a confocal microscope. You don't need to know the specifics now. Foon will get into that later into our program, into how staining works, how imaging works, and we'll have videos of her actually working in lab obtaining data. And then microglia, the cells that we will focus the most on this summer, they're the immune cells of the brain, and they're pretty cool. But cells are pretty awesome, and that there's a lot that we can do with data science and image processing, but it's super underutilized. So I want you to see in the middle of this multi-level pie chart here, there's two types of microscopy: general fluorescent and confocal microscopy. Those are just different ways of looking at cells. And there's three main types of cell groups in the central nervous system. But what we're going to be studying are these glia. Glia are broken up into three main cell types: microglia in the CNS. There are other types of glia in the peripheral nervous system, as well as in like your digestive system. But in the brain, glia are microglia, astrocytes, and oligodendrocytes. But this outer ring includes data science and image processing. And so the number of papers that use microglia and confocal microscope decreases to just this tiny sliver if you actually use data science. So my goal this summer is to train you to be data scientists so that in the future, some of these cell types will have way more papers and research done using advanced data science techniques to study those cells. If you want to learn more about myeloid cells of the central nervous system parenchyma, here's a fantastic paper from Nature. So why did I pick microglia to study, and why will we be studying this summer? Well, this summer we're going to be studying cell morphology, or how cells change shape, and microglia can actually change their shape quite significantly. Over the summer, we'll get into different parts of how they change shape, and we'll use different experimental data in order to explore that. But what you should know is that when they are resting, I say this in like a quotation mark tone, they're actually active. They're highly branched, and these branches move throughout the brain in order to sense what's going on so they can respond. They can respond in many ways. They can retract some branches, grow branches in different directions. They can swell all their branches. They can become completely amoeboid and grow end feet and move across the brain. There's a lot that they can do. They can transition in multiple ways, but they also change their shape based on injury, based on a therapeutic treatment. They have regional and sex-based reactivity. So what this means is that male sex and female sex animals, when we study, have a slightly different way in which their microglia respond to injury and treatment, as well as microglia in different regions of the brain respond in different ways. They're naturally heterogeneous, so even when you have a healthy brain, not all of your cells are going to be in this shape. They will exhibit all of the shapes at one time, just to different extents. And they also change based on species. In my work, in the NANCE lab, we mainly study rats and then secondary ferrets. But in some of my PhD work, I've had the opportunity to study rabbits, pigs. We have a non-human primate brain I haven't gotten to study yet. We have some sheep brains, as well as, I'm missing a species. Rabbits, ferrets, oh, and mice. Mice, a very common animal to study the brain in. So then this is called a Sankey diagram. I just want to introduce you to different data visualizations. As part of the summer, we will be making different visuals. But if you break down the number of papers, so every node equals a number of papers, and every edge is the number of papers that include 2 nodes, you can see how prolific microglia cell immunofluorescent images are, and that they're studied in a ton of different species, and that there are a ton of different features that we can get out of microglia in different ways. Don't worry if you don't know what segmentation and thresholding are now. I will introduce them in a few weeks, and a bunch of different softwares that we can use to study them. But if you look at this features line, the edges are really skinny, which means there's only a couple papers that are actually studying every individual feature, even though there's so much that can be gleaned about microglia. My goal this summer is that you all are able to study a much larger portion of these features just by learning what we have to teach applied to microglia, but you could do this on any cell. So an introduction to like the wet lab side of things. I told you Foon will explain things a lot more. The way I think about my work is like just kind of fitting shapes into boxes and figuring out what that means. So if kids can do this, I can do this. This is an excerpt from our paper with Dr. Tommy Wood, a collaborator in neonatology, on the ferret brain. So in the NANS lab, what we do is we take these images. This is an example of immunofluorescent images of microglia. But these images are obtained from brain slices. So we extract the brain from an animal, we slice the brain, we put that brain in media, which keeps it alive. We put that in an incubator. Foon will describe this entire process. And then we have ways that we can injure those slices, and we can treat those slices, and then we can image their cells. So we did this with the neonatology team, specifically in ferrets. Why study ferrets? We'll be talking about it a lot this summer. So rats are the main animal model we use, as they're a good neurodevelopmental model of the brain, but their brain is smooth or lysencephalic, whereas the human brain is gyrated. So the ferret brain is gyrated a lot more like the human brain. They have a similar gray to white matter ratio. They have more complex social dynamics, and so Tommy's group is really interested in using a ferret model. What you're going to learn this summer is this microglia quantification pipeline. So from those slices, we take all these images and then we study the cells on 2 levels. Individually, we use data science to get a bunch of features like the perimeter, the area. Don't worry about what those mean now. I'll introduce them in 2 weeks. And then on a machine learning side, we study population characteristics, where we ask a machine learning model to say what are the most common shapes that exist, and can you go back in and cluster them for us. Then we use a lot of data visualization and experimental data management in order to split those into groups that actually fulfill a hypothesis. The most common method to do a process like this without coding is with a software called Ph.D. or ImageJ. So there's a paper over here if you're interested in learning more about ImageJ. What that looks like in practice, and you will do this by the end of the summer, I just want to give you a heads up here, is that A on the left is a grayscale version of how we take our images off our confocal. B is after we use a computational process to segment them, and C is when we've gone back in and we've split them into groups and we've colored them, and now we want to analyze how those groups are related. This is just some results that came out of that paper to show you really cool things that we can do, where every color is a different region and perimeter, area, and circularity are different features we can study, and there's 2 hours of injury versus a controlled group, where we can see that microglia change to a different extent across region, or they have opposing trends, and there's just a ton we can get out, and you will have the opportunity to explore data on your own later this summer. But now I want to do interactive session 2. Please forget about the paper. Don't look at the paper when you're doing this. So I excluded the QR code from the corner here. Go back to the exact same form, go on to the second interactive session, and answer these questions. So I want you to just start looking at cells. Don't worry about if an answer is right or wrong. Just look at these cells and answer those questions, and pause the video until you're done with interactive session 2. There's the paper if you want to know the answers. Whew! Thanks everybody for bearing with that. I know that was a lot of information, but it was a quick introduction to a bunch of topics we're going to deep dive a lot later this summer. If you have any questions about those things specifically, tomorrow I'm on an asynchronous office hours, which means that I'm responding within 2 hours on Slack, and I'll be available. If you can hear that, my cat's automatic feeder just went off, but I'll be available on Slack all day to answer those questions and to chat with you all more in a live a more live feeling situation. So we begin learning this pipeline next week. Look forward to it. Please return to that Google form and now complete the Data Science Module 1 reflection sessions for the introduction to the NANS lab. This is important to me as the reflective process is a way for your brain to retain information. If you don't believe that, there's a fantastic 1983 paper from the Journal of Humanistic Psychology that talks about it, and lots of memes that tell you that thinking about what you just learned is sometimes more important than learning it itself. So I'm going to pause. Please pause the video, fill out the reflection, and we'll go on to the second part of this module. Great. Okay, now let's get into the data scientist role. This is a faster part of this module, but the purpose of this is I want you to think about data science in a different way and possibly shift your perspective and get into more flexible thinking. So I want to develop a holistic image of a data scientist: who, what, when, where, how, and why. The way that this is going to work is that you're going to use the exact same Google form. This time I've included the link in this way. And I'm going to present you with two options on the screen, where one will be on an option with a green button and one with a red button. You'll go into that portion of the Google form and you'll select green or red based on how you agree. There's a fun little meme on the side, which is what is a data scientist, and it's Tom? Yeah, Tom from Tom and Jerry, using all these different packages. So priming questions. This is the first section that we're going to go through. The purpose of these questions is to get you used to the mechanics of the activity and to prompt you to start thinking creatively about what a data scientist is. So the first question, please go to that Google form and select green or red to which image you agree with more. When you think of a data scientist, which workspace is more similar to the visual in your head? I also ask you to say why, so please just pause the video until you write out a short statement of why. It doesn't need to be very long. Second priming question. When you think of companies that employ data scientists, which picture relates more to the idea in your head? This picture on the left of a bunch of different phone apps, or this picture of the right of a female presenting person with gloves doing what looks like a pH test next to a microscope? Please select either green or red and pause the video until you fill out your short answer of why. Priming question 3. When you think of who is or can become a data scientist, which image comes into your head? The group on the left, with a bunch of textbooks and just a couple laptops who are clearly working together, or the person on the right who is holding up their hand with a prosthetic arm and what looks like VR goggles? Please select green or red in the Google form and provide a short answer as to why. Great. So those are our priming questions. I hope that you understand the activity better now. Now we're going to get into some main questions. The purpose of these are to refine your image of a data scientist in your head. So the first question, what year was the first written description of a data scientist? 1962 or 1992? Please fill out the Google form. Please don't change your answers. There's no grades in this class. We just want participations. That's the only way that we are grading or judging you on a scale. So please just put the first answer, and if it's wrong, that's okay. That's totally okay. Get used to being wrong. It happens in research. So the first mention was actually in 1962. John W. Turkey described his transition from a statistician to a data scientist and his paper, The Future of Data Analysis. Second question, according to Glassdoor, what is a data scientist ranking on the list of best jobs in America for 2019? First or 12th? Pause the video, answer the question, and check back in. First, in 2019, a data scientist was first on the list for 4 years in a row, according to Glassdoor, based on job satisfaction, openings, and median base salary. It's been sitting at number 3 since about 2020. Next question. What is the most common degree of field of study for data scientists? Computer science or business and economics? Pause the video, choose your answer, and then check back in when you're done in that form. It's actually a pretty close split between the two of them, where you can actually see that natural sciences, math, and non-computer science engineering are other large sections. Those sections are increasing, and the amount that engineering, math, and natural sciences takes up the ratio increases with terminal degree. So it's a larger ratio for masters and an even larger ratio for PhDs. But at the bachelor's level, it's mostly computer scientists and business and econ. Okay, next question. What educational background do you think is the most important to data scientists? Is it the coding languages that you know or statistics? Pause the video, take a second to think about it, fill out the form, and then check back in. It's both. Data scientists are interdisciplinary. There are data scientists who know a ton of coding languages. I myself practice in about 3 now. They know a lot of software engineering practices, or they're data scientists who are mainly into statistics or mainly into learning and bring in their own specialty knowledge area, such as cell morphology, to us. I think it's the really amazing interdisciplinary work of data scientists that makes the role so cool. But it's going to depend on where you're working and what field you're working in. I think this is our second to last question. What types of data do most data scientists handle? Please click green or red on that Google poll based on which one you more connect with. There's no wrong or right answer to this one. I just want to gauge where the group is at. Pause the video until you've selected your answer, and then we'll move on. What separates a data scientist from a data analyst or a data engineer? Click green if you agree with training, or red if you think it's their focus. Pause the video, fill out the Google form, and check back in. Their focus. Data scientists have a specific focus of application in their area of expertise. Other than that, many job descriptions can truly overlap depending on your field. Great, so that was our main introduction to a data scientist and the role of a data scientist. Please go into the next section of that form and fill out the recap activity of how has the idea of a data scientist changed and how so. Pause the video until you've done so, and then we'll get into the last section. Reminder, you can pause this video and leave and return to this module at any point. That's why we're asynchronous this summer. I want it to fit into your schedules. I also want to leave us with this really cool quote. The world is one big data science problem, by Andrew McAfee, co-director of the MIT Initiative on the Digital Economy. Cool. So the last part of our presentation tells me to cut to Google Colab. So we will be working with Google Colab a lot this summer. And so I just want you to get used to Google Colab today. So what we're going to do is I'm going to go with you. Please pause this video whenever you need to show you how to work Google Colab. So you should just be able to open this. And as long as you have a Google account, you should have access to a certain amount of storage in Google Colab notebooks. Notebooks are an interactive development environment, or an IDE, that allows you to code without having to download a lot onto your computer. This works for our purposes this summer. If you're going to code a lot more, then you're likely going to download packages onto your computer and get more complicated, but Google Colab is an incredibly powerful tool that a lot of data science programs and computer science programs use, especially in introductory courses. So up in this corner here, I'm just going to title my notebook Textile 2023 Data Science Module 1. You can title it whatever you want to. You're not going to have to submit this. I just want to make sure that you all are able to work with Google Colab this session. So this is the last part of our module. So first we're going to just write some markdown. We're going to say Introduction to Python. And I'm just going to hit Shift Enter. And when I hit Shift Enter, this turns this in here. But it's in code. So if I wanted text, I could have said add text, and I could write Intro to Python. And when I hit Shift Enter that time, it looks different. You can change this to text. Oh my goodness. Bear with me. I haven't coded since before I I'm just going to delete it. You can swap between code and text. I will look up the shortcut and remember it at some point. But if you write a cell and you mean it to be text, you can change it. Oh, there. Um, but text is what we call markdown. So when we work in notebooks, this is really common with data scientists. We like to mark down our code. So we like to provide a lot of documentation about why we're doing what we're doing. So what I love to do is to add in purpose statements. So our purpose today is to begin practicing and working with Python. Please open Google Colab and write along with me. I'm going to hit Shift Enter again so you can see that. I'm going to add one more line of text. You can go ahead and put that in like the same text box. I put them in different text boxes. It's really going to just depend on your coding style. But documentation, especially as scientists who need to use lab notebooks, is super important. So let's get used to documentation and let's get used to making it look pretty by things like bolding and headers. If you want to learn more about documentation languages, I don't know why that didn't bold it. Is it being weird? Documentation and languages, just being able to do documentation and markdown is a huge part of coding. And so you can look up markdown languages. You can get used to LaTeX or Overleaf, which is what you have to do all computer science homework in most of the time. Those types of things. Now we're going to do our very first coding. So this time we want a line of code. It has this play button to the left, so that's how I know it's code. But if you're not sure, hover up here, add another code box. And all I'm going to say is print hello world. Why is it mad at me? Oops. Oh, it's supposed to be mad at me. I'm following my own instructions. Okay, this doesn't work. Why doesn't it work? It says that we have invalid syntax. So this time we're going to say print, open parentheses, open quotation marks, hello world. I was surprised by my own error, even though my teaching instructions put the error in for purpose. So when we put it in quotations, this turns hello world into a string. And so this is what we're saying. We're using a created command that exists in Python, and we're saying hello world, just return that on the system. It's a string. Why this didn't work before is that hello world was a variable. Since it wasn't in these quotation marks, it was considered a variable. If we did something like say hello underscore world equals a string hello space world, and then we retried print hello underscore world, then it would work. These are just some of the minutia of like coding that you will get used to. I just want to show you that things can happen, but we'll work through them as we go. That's why we have office hours. So you can ask us questions. And that's why we started today. And I want you all getting used to this. We could not have just used hello space world as a variable, as Python doesn't like that spacing context. So that's why I used an underscore. But you could use something called camel case. There's a lot of different ways. Let's go ahead and add in another line of text here, and let's do step 2, print a user-friendly greeting. Please feel free to pause this at any point. I like my steps to be italicized in case you need to catch up. If Google Colab isn't working and you're really struggling, I highly recommend give yourself a time limit and then either message me or Nell Shemek for your questions. We're both of us perfectly able to help you with Google Colab. Step 2, so the first thing we're going to do, so I'm going to use here this hashtag in order to document my code. So this is different than Markdown in that you typically include this within the cell that you want. So if you just put this, it's not going to read it as code. It knows it's some type of documentation. And then if I just hit enter, not shift enter, it lets me go down spaces. So I'm gonna name some username my name. You can use my name or you can use yours. So I'm gonna say user underscore name equals quotation mark Holly. And then I'm going to make another line of codes that saves the variable into the system. So I'm going to say print this string hello, and then I'm going to say comma, and then username. And then next I'm going to say print it is nice to meet you, exclamation point. If I hit Shift Enter, what prints out is, hello, Holly, it's nice to meet you. So you're learning in coding how to combine strings and how to combine variables in order to make statements. I know that everyone in this class has a different level of comfort with coding. That's totally okay. That's why this is module based. If this is really familiar with you, speed through this process and just go to the Google form and fill out, did Google Colab work for you and do you have any questions? If this isn't familiar for you, please replay this as many times as you need. Message me at Nels. Go to that free code camp link from earlier in the video. Get some more practice. Code Academy, as well as a few other places, can also give you that teaching. But don't worry, it will just take time and we will get there with class. Okay, step 3. I'm going to do italics again. So italics are within asterisks. It's giving me a hard time after I print. But step 3, the area of a circle. I like to do this to give you all an introduction to equations. And we're also going to do an introduction to import statements. So what we're going to do here is import numpy as MP. If this part isn't working, please message me or Nels and we'll help you through it. But Google Colab should have this package called numpy already downloaded. So what NumPy is Python is a large coding language, and then there are a bunch of different packages created that work in Python that you can download for free. Google Colab already has them downloaded, so don't worry about downloading anything. But what we need to do, because every time we create a notebook, it doesn't want to load in every single thing that works for Python, we need to tell Google Colab, I now want in this NumPy, I now want in this IPYNB, so in this Python notebook, I want NumPy to now be active. And I want it to be active under the name NP instead of NumPy. This is just because it's a little bit faster and it's so commonly used. You could just use NumPy, but then you'd have to change some things later. So we're going to actually write our first algorithm in code. So we're going to define some radius r equals 3, and we're going to write some equation for area. So we're going to say area equals np, so that's calling numpy dot pi. So numpy in the package has no parentheses, has a set function that works for the number pi. And we're going to do asterisk r, so we can times by the radius, and we're going to do asterisk asterisk 2. In Python, a caret is not an exponent, it's 2 asterisks. So to raise something to a second power, we're going to do 2 asterisks. And then we're going to ask it to print the area that we created. So this should print out 28.27433. If you're having issues with this, please double check that all of your syntax, so the exact way things are aligned and how they're typed out, is correct. Try to troubleshoot for a few minutes. If you're still having issues, message me or Nels. I want you all to get used to being able to message us and asking us for help. Being able to ask people for help is super important in research and in higher education. Oop, I did not make this text. So I'm going to make a text cell, step 3b. I guess we'll say this is step 3a. Doing it in a function. So now we're going to define functions for the first time. I don't use a ton of functions for my part of this summer, but Nels will have more functions working, so I want to introduce you to them. So when you define a function, the definition of the function, what its name is find area. And all of the variables that fit into that are just r. So when we use a function, I'm going to say this function, find area, only has this variable r. And when you do it, I want you to return this equation, np.pi times r raised to the power 2. Now if we said something like we called that function find area and we said that the r value equals 3, it'll automatically run that value, which is pretty cool. Let's add another text file here, and let's call it asterisk step 3c, doing more with a function faster. So in just the way we use that function, it didn't seem any faster than just defining the equation ourselves. But if we had a number list, so let's define some variable number list equals a list of a range of numbers from 0 to 10. you can print number list to see what that looks like. So what I've done here is I've defined some variable number list. I've asked it to create a list. This is a variable type in Python, and I want that list to be the numbers that range from 0 to 10. It's important to know that in Python, we always start with the number 0. So counting starts from 0 in Python. This isn't true for all languages. So we only go to 0 to 9 when you create that range. This is a place where a lot of syntax can get confused, even for regular users. Now we're going to create what's called a for loop. So we're going to say for every number that exists in number list. So this is saying for the number 0, for the number 1, for the number 2, for the number 3, for the number 4, and so on to the number 9, colon. And when you hit enter this time, it should already have a tab. You need that tab. That's necessary for syntax. The area is going to equal find area numbers. So we're going to define this new variable area based on that definition we created, where the computer is gonna fill in for the R variable with each individual number in this list. During that for loop, we also want it to print this string that says the radius, colon, close quotation marks, numbers, so what number was actually put into that variable, and then what the area that was returned was. So the string area, comma, then the variable area. And when you hit shift enter, oh, numbers. There we go. See, even I make mistakes, and I'm reading off of my own thing. For numbers in number list, area equals find area numbers, print radius, print numbers, print area. And then you can run a ton and store them and print them really, really quickly. So that's why using a function and something like a for loop becomes a faster process. The final thing that I'm going to introduce you all to today is plotting, because we have to do a lot of plotting. In order to look at images in Python, we actually plot the images. So I'm just going to introduce you to some basic plotting today. So I'm going to say this is step 4, intro to plotting. Again, please pause the video whenever you need to. You can return to it. You can go over it as many times as you want. This time we're going to import this package called matplotlib.pyplot as PLT. So whenever we call it, we're going to call it by this name PLT, but this is what the entire package is. And this is just the most basic matplotlib Python plotting function. And we're going to use this thing called a magic key. So this percentage sign is magic, and we're going to ask it to do matplotlib inline. And all this is going to do is when we make our plots, it's going to pop them in this notebook in line with our boxes. So to make our first plot, what we're going to do is we're going to define some figure. So we're just going to say fig, comma, ax. There's no way you could intuitively know to do this. This is just a thing that you learn and memorize by coding a lot. If you look up how to do a plot, it will always contain code that tells you to do this. Like, don't worry if this doesn't seem intuitive. It's just the way that the language was defined. So we're defining a figure, and then we're defining axes on that figure using this function from matplotlib pyplot that's called subplots. And then we just leave an open parentheses, and we hit enter. And we say for the axes, we want to set them with these limits. So open parentheses, x limit equals -1 to 1, and our y limit equals -1 to 1. So this is just setting our x and y axes to be from -1 to 1. And then I want to plot a circle because it's a basic shape and we're just teaching the beginning of plotting. So I'm going to name it just as variable a circle equals plot circle. Start at the origin 0 by 0 and make it 0.5 in radius. If you wanted to know how I know how to put these things in, if you look up matplotlib circle, matplotlib circle function, you can get to like a circle, or you can get to a Stack Overflow that will describe how circles work and how you can define them in different colors. So it's just a way that matplotlib defined this circle pattern. You can even look at the code itself and see, oh, you need some XY, you need some radius. This is just a way to look into code. Don't worry if that stresses you out. We'll get more practice with it. And then I'm going to use this axe add artist a circle. This just makes it look nice. Did I do wrong? X dot set x lem equals -1 to 1 y lem. I forgot the equal sign. Yay, now we made a circle. Coding and small syntax errors are really common. This time I just want to get you more practice with using a for loop and using circles. So we're going to say 4 numbers in that numbers list before. So this is going to go back to that list of numbers we defined, and it's going to loop through each of the numbers individually. I want you to create a figure. So fig ax, it's almost the exact same thing as before. Make those subplots if I spell things correctly. And then ax set x lim equals, this time let's make it bigger because we're trying a bunch of different variables. So let's do -10 to 10. Y lim equals, don't forget the equal sign like I did. And let's define some circle is plt circle. Always we want it to start at the origin, so that's 0, 0. But this time we want it to use our numbers variable divided by 2. Otherwise it'll get too big. And then ax add artist a circle. If I spelled artist correctly. A circle. Meh. Meh. It's number list. Wow, I'm really getting myself with, so numbers in number list. Getting myself with plurals today. And when you do that, you'll see that we're creating a series of plots. So we have this 0 radius plot, we have our small radius of 1, and then we're increasing to create these circles. And that is an introduction to Google Colab, is an introduction to Python. Go through it as many times as you want to. Please message me in Nels. Please go if you have any questions. I'm online for office hours tomorrow, and please just go ahead and go into that Google form, fill out the rest of the questions. Definitely, if Google Colab is giving you issues, message me or Nels as soon as you can. We need to get that figured out for the summer. But if everything was good, then you're good to go. And we will have our next module next week. Thank you so much, everybody.